Hi, my name is John, and with me is Dave, who will be making video presentation for this second part of the video presentation. For me, I'll be talking about the CBC, the NMJ, and lastly the ECC, whilst Dave will come to give the concluding part of it. Now, to start with, I'm talking about the CBC with sliding filament theory. The sliding filament theory describes the mechanism that allows muscles to contract. According to this theory, myosin binds to actin. Then the myosin then alters its configuration, resulting in a stroke that pulls on the actin filament and causes it to slide across the myosin filament. The overall process shortens the sarcomere structure, but that does not change the actual length of either filament. Then the function that enables the release of energy from ATP, the myosin heads attached to the actin filament bend to pull the actin filaments closer together, then release, attach, and pull again. Energy from ATP is therefore required for the myosin head to release from the actin filament. Otherwise, the myosin head will remain in the same place, and the muscle, of course, wouldn't contract. Even though the filaments are moving, the filaments themselves never actually get shorter or longer, not minding the movement that is apparent. When ATPs are depleted, myosin becomes incapable of detaching from actin, and the muscles remain in a taut, flexed state. This is the cause of rigor mortis that we talk about everywhere and every time. When is ATP required for muscles according to the sliding filament theory? It is required, of course, in this process, and this is evident when myosin head will hydrolyze the ATP to ADP plus PI, being bound to ADP. It allows the myosin head to form cross bridges by binding to actin. As ADP detaches from the myosin head, the head will produce the power stroke motion, where the myosin heads will rotate toward the sarcomeres. The myosin head will be locked in this position, and it attaches to the actin until another ATP molecule comes and attaches to the myosin head. This will allow the head to detach from actin, and of course it will reorient itself to complete the process again. So the process actually does not end at that first shot, it has to be repeated. That is all about the first part of my 3 parts. The second one I'm going to talk about is the NMJ. Of course, the almighty and most popular for me, the neuromuscular junction. Of course, this is basically the point the neurons speak to the muscles to tell that muscle you have to do something, and that thing is nothing but to contract. So the neurons actually speak, and the muscle also hear, and when they hear, they respond by contracting. In this case, the neuron is a motor neuron, while the muscle has to be the skeletal muscles, knowing full well that we have different kinds of muscles. So I'm going to look at it from 7 stages that it really works, and the things that are obtainable in these 7 stages. The first is the action potential travels down the axon to the axon terminal. Now, the second stage comes where the electrogated calcium channels will open and calcium enters. Then the axon enters into the axon terminal. Now, the third stage is where calcium causes the vesicles to release ACHG into the synaptic cleft. This is just the opening. That is where it enters. The calcium enters the synaptic cleft, the synaptic opening, if you like. Then, in the fourth stage, the ACH diffuses across the synaptic cleft and binds to its receptors on the muscles. Then the fifth stage is this ACH binding opens ion channels, and sodium, of course, will rush into these muscles. If these muscles reaches the threshold of minus 55 MV at the motor end plate, and AP occurs on the muscle. And finally, ACH is broken down by acetylcholine, ACH. Now, I want to talk about the last stage, which is ECC, excitation, concentration, coupling. Excitation is just the stimulation by neuron, while contraction is the interaction between myosin and actin. Then I'm going to look at it again on 6 stages. The first one is the end plate potential, when it is produced and what happens after the end plate. Of course, the action potential, where it goes through the T tubule, the release of calcium, concentration, and calcium pumping. Now let's look at it, the processes. At the end plate potential, the production of it, ACH is released by motor neuron, and it binds ACH receptors. It therefore depolarizes motor end plate and causes an electrical phenomenon known as an end plate potential. Then this end plate potential will therefore trigger formation of action potential, which travels across the sarcolemma. And the action potential is propagated down the T tubules. Action potential also triggers release of calcium from sarcoplasmic reticulum, which is PR. Then comes the presence of calcium, which triggers contraction. It would be right to say that there is no contraction when calcium is not present. So the presence of calcium actually makes contraction to occur. And finally, I'm also going to see that the action that is produced keeps pumping. So contraction therefore continues until calcium is pumped back into sarcoplasmic reticulum. Thank you.
All right, now for the second part of the presentation, I'll be talking about the dead fish and the raw chicken and the dead fish in the rigor mortis process. Okay, for the explanation of how a dead fish or a raw chicken meat can still move even after being beheaded, I've came down to 3 factors that can actually stimulate such a reaction in the muscle. And those factors will be ATP reserved in the body, sodium concentration, and neuron stimulation. So for the ATP reserve, all living organisms require ATP as a source of energy for cellular function, which can be obtained from photosynthesis, cellular respiration, anaerobic respiration, along with proteins and fat. Carbohydrates are one of the 3 nutrients found in food and drinks. The body breaks down carbohydrate into glycogen in the small intestine. The body breaks down carbohydrate into glucose in the small intestine. Then glucose, and after the body have used the energy that it needs, the extra glucose and the extra energy that the body is not going to be put into use at the moment, the body stores those as fat in the abdomen and under the skin as glycogen. And in order to get the glycogen, the glucose that are not being used will have to go through the liver and go through a process and turning into the fat that's going to be stored under the skin, or only known as the subcutaneous layer. Um, for the second aspect of why the meat will be moving, I have also sodium concentration. So when we're cooking fish or chicken, we all know that like you have to put the meat in salt in order to either preserve it or for pre-seasoning purposes so we can have a better taste. And that salt, upon laying on the meat, would actually diffuse through the skin by the process of diffusion, where the small sodium ion will penetrate the skin molecules using, like the meat juice that's going to be drawn out by the concentration of sodium on the outside. And once the sodium is mixed with that lipid or juice or water, whatever it's gonna be, the skin is going to reabsorb that water with the concentration of sodium in it. So when the fish or the chicken is killed, sure they're dead, but the neurons don't stop working right away. Since the creature is dead and the nervous system can't send neuron signals for action potential, salt acts as a substitute. The concentration of sodium send sodium ion into the cell, which cause the cell to open up, which will react to the membrane potential due to the reserve of energy that the cell has. The neuron cell membrane is partially permeable to sodium ion. So sodium atoms slowly leak into the neuron. In order for the cell to maintain a negative resting membrane potential, it has to pump that potential back in the cell and sodium out, so the cell can, at the same time, so the cell can maintain its own balance. And for the third aspect, we have neuron activation, which I also like to call the trickery. So for this one, the concentration of sodium will actually trigger the neurons that are still active and cause them to fire, signaling the muscle to actually contract, using the reserved energy in the body stored as glycogen until the body run out or the stimuli stop. Unable to release contraction, all the muscles of the body remain tense, causing rigor mortis, which is something that John mentioned in his first part of the video, which is a biochemical change that involves 4 stages. First, we have autolysis, and then we have bloat, we have active decay, and then we have skeletonization. Overall, the actin and myosin become stuck together, so kind of like put the muscle in a state where it cannot relax. So for the first one, which is autolysis, autolysis is the process where the cell and the tissues of the body actually get destroyed by using their own enzyme, which is most often from the lysosome. And for bloat, as it mentioned, it is pretty self-accurate. As bacteria decompose in your body, they release gases that cause the body to bloat, which is in turn force ooze of fluid out of your body. For the third, we have active decay. So for this stage, after decomposition of all the internal organs, most of the lipid in the body have gone, so the skin starts to turn like darker and darker. And finally, we reach the stage of skeletonization, where also pretty self-explanatory. It is literally where you have your bone left behind after the decomposition process have taken place. Thank you.